Breast cancer affects a multitude of women in America. About 5 to 10% of breast cancer cases can be attributed to hereditary causes, and genetic tests can test for 80 to 90% of all hereditary breast cancer. There are around 22 different genes that have causal mutations thought to increase the risk for breast cancer, but these are very rare and only account for about 2 to 5% of breast cancer cases. There are 5 main genes with SNPs believed to have risk alleles for breast cancer. Arguably, the 2 most significant of these are BRCA1 and BRCA2, which are only expressed in breast and ovarian tissue. Both of these genes code for proteins which act as tumor suppressors, so they pause the cell cycle to avoid dividing too quickly. Mutations of these genes can lead to the development of breast cancer because if the tumor suppressor genes can't work effectively, then diseases like breast cancer can spread. Our SNP chip tests for 7 variations on the BRCA1 gene. Out of these, the 2 most reliable predictors of risk for the development of breast cancer are these 2. The first SNP has 2 key causal genotypes, which both have been proven to indicate a high likelihood of developing breast cancer, and they are given magnitude ratings of 7 and 6 respectively, which mean they have been proven to be highly associated with the disease. The normal C allele codes for alanine, and the mutation A allele codes for glutamic acid. For the second SNP, there are 2 alleles that are also linked with breast cancer, as their magnitudes are both 6. The normal C allele codes for the arginine, while the mutation T allele codes for tryptophan. BRCA1 is autosomal dominant, meaning one allele that codes for the mutation is guaranteed to cause the mutation, like these 2 genotypes. So, if the test reveals prevalence of the mutated copy with the rarer alleles, the tumor suppressor genes cannot function effectively.
There are some limitations to our test. For one, a scientific study that focused on over 200 individuals undergoing genetic testing found that those who tested positively for predisposition of the targeted disease developed heightened levels of anxiety and depression compared to their age group who tested negatively. This is doubly revealing regarding children, as when they are tested early in life, anxiety and depression then can follow them for the rest of their lives. Likewise, this could happen after undergoing our genetic test for BRCA1. While there are strong correlations between certain types of genes and the risk of developing breast cancer, there is no complete understanding that considers all possible risk factors. While all the SNPs on our chip are associated with the disease in varying levels, and an individual's outcome from having the SNPs varies based on other genetic factors. The best way to determine a family's risk of breast cancer is to test the SNPs of a family's members who have had breast cancer and determine which ones were contributors. Also, a person who tests positive for specific SNPs associated with breast cancer development may choose to get a mastectomy, which can be financially and physically tolling, and she can't even be sure she would have developed the disease. Also, if a person tests positive for related genes and the patient is past the age where she is healthy enough to pursue surgery, she and her family then have to deal with the emotional aftermath of a potentially terminal illness. Ultimately, the individual is responsible for weighing the negatives and positives of the test to make an informed decision.